a series of filtering steps to identify a strict set of orphans of species-specific genes. We're in bacteria right now. I should say that. So these are proteins that come from bacteria that live in the human gut. Okay. So out of this starting set of 4.7 million protein families, we kept 630,000 single species clusters. So these are orphans. These are sequences, proteins that you only find one species and nowhere else. And so this was just to describe very briefly the data behind this. And this paper is on, and there's a preprint of this paper that's still under review if you're interested. So now, remember, the idea is to try to, using machine learning, find a pattern within non-statistically significant blast hits that would imply that a gene that does not have any significant blast hits has evolved via sequence divergence, right? So we built our positive and negative sets in the following way. So for the positive, we just did very basic simulation using a couple of different models and a couple of different parameters, which I'm not going to go into. So we took one of these proteins that were not orphan, non-orphan, so conserved in the data set that I just showed you, right? And we simulated them to the point where they became, they didn't look like any other protein in that data set. And for the negative set, we just flipped the protein sequence, so reading it from in the other direction. So by doing that, you get something that is completely unnatural while keeping a natural composition. So evolution doesn't work in this way. And so when you reverse a protein sequence, when you then go to blast it, when you go to look for sequence similarity, you wouldn't find any true hits. That's something that's been established. So we constructed these two, we started with these two sets of sequences. Then we did a round of similarity search in the initial protein catalog that I mentioned. And then we filtered out all the proteins that had statistically significant matches. So the ones that were not really species specific, let's say. And we kept only the ones that had garbage, blast or diamond. So I should stop saying blast. We've used diamond for all of that. If some of the people that are involved in developing diamond are in the audience, I'm sorry about that. It's actually diamond, which is a great tool. There isn't anyone, right? Okay. So by doing that, we have a balanced set. So we have equal positive and negative labels, and there's 80,000 orphan sequences. And so we just trained a few simple classifier models on that, using like a standard cross-validation and leaving out and testing on 20% in the end. And so that's what we got. These are the classification that we get. So some of the lines are not showing up in the PDF. But that's fine. You can see that the main point is that we got the best accuracy that we got is 0.8, which is not too bad for a simple first try at something like this. Now, I don't know if maybe there is a paper from 10, 15 years ago where someone tried this exact same thing, and so we're not really reinventing anything. And if there is, please let me know. But I think from what I know, this is like a kind of just a first try at this. So it seems like there is some sort of pattern within these blast diamond matches, diamond hits that we would throw out that can discriminate between these orphan genes that we have simulated, like by applying this divergent evolution, and these other ones that were just flipped. And so they shouldn't have any true similarity match. And so the last thing, and I still have plenty of time. That's great. Story is a lot simpler than I thought. And so the last thing is that then we looked at which features came up as the most important. And I didn't explain that, but the way we did like a simple featureization thing. So we just took the different columns from the diamond output, and we turned them into some simple features, which include the total query coverage, the average e-value, the minimum e-value, the highest alignment length for a hit, the highest bit score, etc. And it looks like so I should say for the Bayesian classifier, right? It's the one that we tested 4 models, and the Bayesian was the one that performed the worst. So we can maybe keep that one out for now. So it seems like query coverage is a fairly, if not the most important feature. So there's this blue bar here. It's the first most important feature in the random forest, and in the logistic regression as well. And then in the boosted trees, it's the second most important. So it seems like there is something there. There's a trace that remains, even though we would not look at it if we just did what everybody does, which is apply an e-value cutoff and then just keep anything that is below that. And so, oh yeah, there's another thing. There's more. So we then took these models and we applied them to a real set of orphan genes, the ones that I mentioned before, that I generated through these filtering steps. And these are the ones where we truly, really don't know where they're coming from. And so we used our classifiers to predict, and we found that there is, again, only a small percentage that is in the divergence class, where the classifiers predict this is how these genes originated based on their patterns in their blast matches. Apart from the Bayesian, but we established that this doesn't work as well. So interestingly, this seems to agree with the synteny-based approach that I mentioned in the beginning of my talk. That one we applied to vertebrates and fungi. So this is bacteria. There's entirely different modes of evolution, but still, it's interesting that the two agree. So that's the end of the story. As I said, this is like a side project, and it's the first try. I think there's a lot more we could do on that. I'm happy to hear your thoughts. And these are the people, as I said again, Yori and Emilios who did the work, and Anne from Wageningen, who we collaborated on that. And thank you all for your attention.